can't hear you. No.
How about now?
Yep.
Yeah, now we can hear you.
All right, I think we're in business. Very good. Okay, so we're going to go over the quiz. I will check answer keys and post scores for everybody a little later on today. But we're going to go over the quiz right now. Then I'm going to probably take about 10 or 15 minutes, finish up talking about proteins. If you can stick around, terrific. If you can't, I will record that part of class today. So hopefully the Zoom will record the whole thing, so you'll have that to use to study for Thursday. Okay, so let's go over this. So I think what I want us to do, because none of these have A, B, C, D, or E on them, let's just pretend they do. So A is at the top, E is at the bottom. So which of these sequences correctly reflects the process of science?
C.
C.
C or B?
C. C.
You
Okay,
can't
so-
prove a hypothesis.
Can't prove a hypothesis, exactly. Okay, so either support or refute a hypothesis. So that's the warning that I told everybody about when we were talking about scientific method, that I was going to put it on the quiz and I'll put it on the test on Thursday. Please don't fall into that misconception that you can prove a hypothesis, that you actually support or refute hypotheses. Okay, so a little story here about growing hot peppers. And so, which of these is the dependent variable in this experiment? A, B, C, or D?
D. D.
So what do you, what
Is
so
it
the,
the number of seeds in each pot?
so you're growing peppers, some are on a heated mat, some are not on the heated mat. What are you measuring?
The temperature.
Okay, so you could check the temperature, but what do you want to see if the temperature makes a difference? What do you
The time.
Time.
Yeah.
time it takes the seeds to germinate, right? The time it takes for the little pepper seeds to appear. What's the independent variable here?
It would be the
Temperature.
heating.
A, B, C, or D? The independent variables on the list here. Which one is it?
A.
A.
It's going to have to be the presence or absence.
Exactly. It's the presence or absence of the heating pad. That's the difference, right? So the difference, the manipulation is the independent variable. The thing that you measure, in this case, the time it takes the seeds to appear, or the seedlings to appear, is the dependent variable. All right. What's the control in the experiment? A, B, C, or D?
B.
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 The seeds on the counter without the heating pad? Yes or no?
Yes.
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 Right?
Yes,
So
sir.
that's the condition that's held constant. Right? The thing that you're comparing. All right. And so what's the hypothesis that's tested here?
Temperature. Oh, A. A.
Temperature affects germination rate, right? What is B?
Prediction.
Prediction, right? And seeds need warmer temperatures to germinate. That depends on the seed, right? So that's not even a choice here, right? So temperature affects germination rate. That's the hypothesis. All right. Which of the following correctly lists the major groups of biologically important macromolecules? A, B, C, or D?
D.
D.
Everybody should get this one right. Okay, good. D. Which is true of a polar covalent bond?
B.
The electrons in the bond spend more time around the other nucleus than the other.
Yep, B. Okay, that's the example of a polar covalent bond. Good. Okay, which of the following are chemical bonds that rely on the attraction of positive and negative charges?
E.
Ionic and hydrogen bonds.
Ionic bonds and hydrogen bonds. So in this case, the E is the correct answer. So ionic bonds, you have two fully charged ions, a negative and a positive, attracted to each other. So pretty strong in the absence of water, fairly weak in the presence of water, but still not as strong as covalent bonds, regardless of the presence or absence of water. Okay? Remember, covalent bonds are a sharing of of a pair of electrons. All right, which of the following holds the hydrogen atoms to the oxygen atom in a molecule of water?
Polar covalent bond.
Polar covalent bonds, right? So the electrons spend more time around the oxygen compared to the hydrogens. That's what makes water a polar molecule. And because water is a polar molecule, it's able to, individual water molecules associate with other water molecules through adhesion or cohesion. Cohesion. And what kind of bonds facilitate cohesion?
Hydrogen bonds.
Hydrogen bonds. Good. What's adhesion?
Water like connecting to like a leaf or something.
Water connecting to, attaching to some other polar molecule, also by hydrogen bonds, right? But so cohesion is water to water. Adhesion is water to some other polar molecule, both of which take place through hydrogen bonds. Okay, good. All right. Which of the following statements about dehydration reactions, also known as condensation reactions, is true?
A.
A. The assembly of complex carbohydrates, triglycerides, and phospholipids depends on this kind of reaction. What are the other two described?
Breakdown. Yeah.
Hydrolysis reactions, the breakdown, right? The breakdown of lipids or of proteins or of complex carbohydrates. Good. All right, a bit about pH. A solution with a pH of 6 has how many hydrogen ions compared to a solution of pH 3?
B.
1,000 times fewer.
1,000 times fewer, right? Because remember, the pH scale is a logarithmic scale. It's a factor of 10. And so the higher the number, the fewer, the lower the concentration of hydrogen ions. So 6 has a lower concentration than 3. How much? Threefold less. 10 times, 100 times, 1,000 times fewer than a solution with a pH of 3. Another question about pH. So we've got two different solutions. Which is true of solution A?
B.
B? Okay, so
No, D.
I selected solution A is more acidic and lower pH.
Yeah, it's D.
what do you think? So solution A has got more hydrogen ions. So is it more acidic or less acidic than B?
More
acidic.
More acidic.
Than B, right? And so is it higher or lower on the pH scale?
It's lower.
Lower. Okay. So that's the understanding the relationship among the pH scale, and whether something is acidic or basic compared to something else, and hydrogen ion concentration. All right. Question 13. Which of the following function mainly as energy storage molecules?
B.
B.
B, complex
B.
carbohydrates and triglycerides. I mean, technically, all of these can be used as an energy, as a source of energy by different pathways. But complex carbohydrates and triglycerides, their primary function of many of them, especially triglycerides and starch and glycogen, are energy storage molecules. All right, what's that thing? What is that molecule?
Fatty acid.
Is it a fatty acid or is it a fat?
Fatty acid.
Right? It's a okay. So what's the difference? If you put fat, why didn't you put fatty acid?
So a fatty acid is the carbon and hydrogen chain. So 3 fatty acids and a glycerin molecule make up a fat.
Very good. Excellent. Yeah. So each of these 3 chains, these hydrocarbon chains, are in fact fatty acid chains that are connected to the glycerol that make up the triglyceride or the fat or the oil. If there were double bonds in here, it would probably be an oil. So that whole thing is a fat. The side chains are the fatty acids. Okay, so don't get confused on that on the test, because I'll probably ask you something very similar to it next time as well. The whole thing is a fat. The chains are fatty acids. Where else do you find fatty acid side chains, and what kind of lipids?
Phospholipids.
In phospholipids, right? But instead of phospholipids, instead of having a third fatty acid side chain, they have that polar head group attached by a phosphate group. Okay, good. Question 15, what are we looking at there?
Complex
C.
carbohydrate.
Complex carb.
Carbohydrate.
Complex carbohydrate, right? Because there's a bunch of sugars attached to each other. What's another name for complex carbohydrates?
Polysaccharides.
Polysaccharides. Good. And what's the special kind of covalent bond that's attaching the sugars to the whole thing?
Double.
Glycosidic
bond.
Remember, there's those three kinds of special
That's it.
three kinds of special covalent bonds that we learned that are important for holding together macromolecules. One for complex carbohydrates, one for some lipids, and one for proteins. What's this? Glycosidic linkage. Good. What's the special covalent bond that holds amino acids together in proteins?
Peptide bond.
Peptide.
Peptide bonds. Good. And what's the bond that holds the fatty acid side chains of the glycerol in triglycerides and phospholipids?
Ester bonds.
Ester bonds. Good.
Ester bonds.
Ester bonds. Very good. Okay. Last one. Functional groups. Which one acts as a weak acid?
Carboxyl. 4.
4, right. The carboxyl group. Good. What is 5?
Hydroxyl.
Hydroxyl group. What's 3?
Methyl.
Methyl.
Oh, methyl.
Yep. What about 2?
Amino.
Amino group. And number 1?
Phosphate.
Phosphate group. Very good. All right. So that's that for the quiz. So now let's